Hi, I'm Giorgio Basile, and today I will tell you the story that we recently published about the role of pancreatic elastase in regulating beta cell homeostasis in type 2 diabetes.
The pancreas is a vital organ that contains cells important to regulate main metabolic functions of our body. In particular, it hosts endocrine cells, grouped in mini organs called islets of Langerhans, that include hormone-producing cells, like the beta cells, that release insulin to maintain normal blood glucose levels. The islets are surrounded by exocrine cells, mainly acinar cells, which produce enzymes important for the digestion of micronutrients, as the pancreatic elastase coded by the SILA genes. However, the expression levels of these enzymes, mostly SILA3B, are elevated in the acinar cells of patients with type 2 diabetes, and through mechanisms that are yet to be defined, it deposits in the islet microenvironment, mostly at the plasma membrane of the beta cells. But what are the effects of the elevation of pancreatic elastase levels in the islet milieu of the beta cells? To test this, we employed human beta cell lines which exert proper beta cell functions. They are able indeed to secrete insulin in response to glucose stimulation. However, the exposure to pathological levels of pancreatic elastase results in the reduction of glucose responsiveness along with a decrease of beta cell viability.
And so, based on this data, we hypothesized that the specific blockade of pancreatic elastase activity in the islet milieu by using small molecules would blunt the detrimental effect of the enzymes and overall increase beta cell viability.
To this end, we carried out high-throughput screening assays, screening more than 16,000 compounds in order to identify potent pancreatic elastase inhibitors. Our major hit was telaprevir, or VX950, a small molecule developed by Vertex to treat patients infected by hepatitis C virus. We then confirmed that telaprevir was very specific against human pancreatic elastase, with limited effects against other proteases. To validate the mitogenic effect of pancreatic elastase on beta cells, we administered telaprevir to human islets in cultured and transplanted in humanized mouse models. Both approaches revealed that telaprevir-mediated pancreatic elastase inhibition promoted beta cell growth and function. To identify the molecular mechanism that linked pancreatic elastase activity and beta cell homeostasis, we combined single-cell RNA sequencing and phosphoantibody macroarrays on human islets incubated with telaprevir. From the following analysis, we identified the protease-activated receptor, or PAR, pathway and the integrin focal adhesion pathway, collectively grouped in the mechano-signaling. These two pathways were potential mediators of the action of pancreatic elastase controlling beta cell growth. Finally, we went back to the human beta cell lines to validate our results. We treated control cells, expressing functioning PAR2 and mechano-signaling pathways, with telaprevir in combination with pancreatic elastase, confirming the mitogenic effect of pancreatic elastase inhibition. However, cells in which PAR2 pathway was blocked by using a specific PAR2 inhibitor and the mechano-signaling pathway was impaired following silencing of the focal adhesion kinase gene are not able to respond to the drug, leading towards an increase of cell death levels.
In conclusion, our data highlight the role of pancreatic elastase in regulating the communication between the exocrine and the endocrine compartment of the pancreas in type 2 diabetes and point at inhibitor of pancreatic elastase activity as potential therapeutic agents to counteract the progression of type 2 diabetes. Thanks for watching this video, and please follow our publication for further details.